Welcome back, everyone. Next up is Bioxytran Inc. They are an OTCQB company trading under the ticker symbol BIXT. The company is a clinical stage biotech with 3 revolutionary platform technologies that were quickly developed using the power of AI. They have a broad spectrum oral antiviral drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that not only neutralizes COVID-19, but also neutralizes influenza and RSV and prevents it from entering cells. They are currently recruiting patients for their dose optimization study, which will give them the ability to proceed with their registrational trial in India after they finish. They have a second platform drug being developed to treat stroke, which is supported by an FDA-approved device that detects tissue oxygenation. And due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. And there is a third platform technology that is capable of treating cancer metastasis. So David, welcome back. Your last presentation on emerging growth was very informative, and I see that you added some new slides to your presentation. So without further ado, the floor is yours, and when you're done, we can take some questions. So jump in whenever you're ready.
Very good, I'm ready.
All right, after you.
Okay, thank you for having me. In Bioxytran, which is biological oxygen transfer, that's the name of the company, we're dealing with complex carbohydrate chemistry. As a public company, we have a forward-looking statement, and we'll start with the first idea or slides, what we do in the company. So there are 3 distinctive platforms that we're working on. The first one is the viral infection platform. The second one is in cancer, and the third one is by oxygen transport. So the underline of doing and dealing with those platforms or making drugs for each platform is a carbohydrate polymer, which deliver in case of oxygen, in case of oxygen transport, deliver oxygen, of course. And the concept is that the carbohydrate or the carbohydrate recognize galectin, which are specific receptors, protein receptors. If you look on what we did in this area, so in the first platform, we created a drug that it's all of it's a complex carbohydrate chemistry, which show a complete inhibition or elimination of the under PCR, of course, of COVID-19 in patients. It's under FDA with an IND. The second platform, it's for cancer metastasis. We're using the sugar or the complex carbohydrate to prevent the spread of tumor or metastasis. We're talking about late-stage cancer patients. And the third one, by the way, this platform, there are clinical trial, and it's a whole package that is underneath that. And the last one, which is the third distinctive platform, we're creating a blood substitute that actually it's an oxygen, universal oxygen carrier that deliver, it's an IV solution that injecting, once inject, it carry oxygen to tissue the same or similar to red blood cells. So to understand where I'm coming from, as a researcher in the late 80s and the beginning of the 90s, I expressed the gene for galectin, and I call it galectin. In my 2 articles, I extract the structure, the chemical structure, the 3-dimensional structure. And since then, there are 10,000 scientific publications published on the receptor, which I called galectin, which is recognized as sugar galactose. This is incredible as a personal note, but the question is how you utilize that type of receptor in medicine and make drugs to fight disease. So let's talk about the first of all, what are galectin? Make sure that we understand that. So as I said, it's a protein receptor which recognizes sugar molecule predominantly, and most of them are responsible or involved in chronic disease. So from that area of galectin, we decided to, one of the places that we can actually show very strong activity is making an antiviral drug, since the galectin appears on all viruses. So if you look on this slide, you see that most viruses require a galectin receptor to enter to the human cell. Secondly, the galectin antagonist can block that entry. And what we do, we basically interfere between the recognition between the virus and the human cell. And around that, we have the intellectual property on the structure of the carbohydrate to interfere, which we call an antagonist to the galectin. There's several galectin on each virus, so unique galectins. So how we do that? If you look on this slide, we take the galectin outside the cell, we make it, and then we make the carbohydrate, and we try to recognize, based on the chemical structure of the protein, which is the galectin, we make the carbohydrate to fit and recognize that specific galectin. By doing that, we actually creating kind of a plug or a way to, which is the inhibitor, to a specific galectin. That's done by NMR studies. And we're doing that, by the way, University of Minnesota, chemistry department, and in collaboration. And as a consequence, after we did that, we know that we have the right structure for the recognition. We manufacture that, doing all the testing, preclinical and all that, animal toxicity and all that. And the next thing, we did clinical trial. And we have the results as summarized here are really showing how accurate you can be, or we can be, in blocking those viruses from recognizing the human cell. In this case, in clinical trial, we have a 100% read by PCR after 7 days. Actually, in 3 days, most of you see 88% or 90%, the patient were out of symptoms, and basically the PCR show negative in 7 days. If you compare it, we didn't compare it in clinical trial, but you compare it in a known drug, which exists today for COVID-19, Paxlovid. We're talking about the Paxlovid has 50% according to the report, and takes 14 days for 50% of the people to get rid of the virus. So the conclusion from that, it's very simple. We have a very strong technology. The question is, since it's phase 2, the question is, should we continue with that in COVID, or we expand to for other viruses? In this case, let's understand what we have. We develop a drug, call it prolactin M, by mouth. It can go to any virus, any coronavirus, any mutation. Doesn't have a limitation since toxicity was minimal, or there was no toxicity in any of the patients. We can expect it will go for many different age groups and so on. And the efficacy was obvious, as I described. So what next? We took the concept of that development, theoretical development by NMR, and designing, it's basically medicinal chemistry, and designing the complex carbohydrate, and we designed against a different galectin fold on that specific virus, which is a herpes virus for specific lesions that exist on the skin. And again, we show result, we publish them, that we can eliminate on the skin the virus. We try other viruses. For example, we try on influenza, H1N1. We did experiment. Those are in vitro experiments. They are not in human, because it's each one require substantial amount of money to go from one virus to the other. But as we use the COVID-19 as a case study, but you can see here that we can do the same. We did the same in influenza. And this one is H1N1 in vitro. And we did the same thing in other viruses, which is parallel to, for example, in the recent news, we have the H5N1. We also, theoretically, we can go after this one, because the H1N1, which we did in vitro, is very similar to the H5N1, the bird flu. So to summarize that, we're talking about something which is similar to antibiotics in the beginning of the 20th century, when antibiotics were discovered for herpes, for syphilis, and later the many type of antibiotics for different bacteria. Here, we opened the door to design different carbohydrates to eliminate different viruses orally or IV, depend on the way we do it, orally, IV, or on the skin. And those, the idea that vaccine is the only way or the most efficient way, actually, we demonstrated we can go after influenza, and you can look in the number of people that have every year influenza and shingles, like on the skin, what we did, and all the respiratory RSV, we have result in RSV, and conjunctivitis on the eye, and of course, long COVID, we can do it IV or orally. So that's the platform that we are after, and we probably in clinical trial, we will redirect ourselves to the best outcome once we find the right partners to move on. The second platform, which is the blood substitute platform, is based on the idea that when there's a patient who gets a stroke, it takes time to get to the hospital to identify if it's hemorrhagic or ischemic. And that time damaged the brain. It's around 1 to 2.5 hours. By that time, the brain can be seriously damaged, lack of oxygen. And what we did, we designed basically a molecule called BIXT25. And that molecule is very small. It's about 5,000 times smaller than red blood cells. What we did, we took the way we do it, we took the erythrocyte, red blood cells, and we took out the hemoglobin out of the red blood cells, and out of the hemoglobin, we took the heme. And by taking the heme, we put the heme on a carbohydrate. And now we have a heme carbohydrate. And as a consequence, we show that we can do it IV. You don't need blood type. It doesn't have immune response. It's stable at room temperature since it's mostly carbohydrate. And for 5 years, we say 3 years, but we tested now for 5 years. And we can make it, if we want to make it a powder. We didn't try to reconstitute as a powder, but that's a possibility. So and we also tested in animals, showing that we can oxygenate animals in a model of a stroke model. And we basically compare it to other drugs. This is a unique position because we are coming right in the ambulance, in the first moment that we see a patient. We can inject that solution to save the brain. Since the molecule is smaller, it very small, it penetrates through the clot and actually can oxygenate the other side. If the brain is hemorrhagic, it means that it has a large amount of blood. It will not hurt the person if we inject that material. So that's very nice to intervene in early stage. How we know that we actually oxygenate the brain? For that, we developed a device that was approved by the FDA. We call it the MDX viewer. And that device actually measures the amount of oxygen every cell used by the mitochondria to create the energy, or the molecule ATP. So we can measure specifically every single molecule of oxygen that is used by the brain. So we can actually track the hypoxia level of the lack of oxygen in the brain, and that's our ability to approve the drug, since we are not really asking for efficacy in terms of medical claim. We're talking about functionality of oxygenating the tissue of the brain or other tissue. And that's how and the underlying development, that device, the underlying development, how we can make it a blood substitute or molecule that can deliver oxygen to the brain. And the last platform, which we do, it's cancer. This is a rich platform that we develop. At least we have a couple of drugs, but the one is for human trial. We choose one of them to neutralize cancer metastasis, the spread of the tumor. So to date, over the years, I developed, and I was involved in my previous venture and by myself, in 140 patients was dosed in 9 phase 1, phase 2 clinical trials. And results are published articles. And complex carbohydrate chemistry as a way to block cancer and cancer metastasis, it's a proven thing. And we just need to take one of the most efficient drugs that recently we designed to take it to human trial, which, by the way, we went all the regulatory phases, and we are ready to go to phase 1/2. In this one, we have a drug that we need to establish the pharmacokinetic parameters, which in the end, recently we did it. And basically, we can have the drug in human once we have the funding. The last thing I want to talk about is the development pipeline, what we are doing now. Since we're doing the pharmacokinetic, we finished the phase 2, but we need to do pharmacokinetic, since we don't know how many tablets a patient need, we gave the maximum. We assume it actually less than the amount of tablets we gave today to patients. So that's part of the trial we're doing. We also want to expand to other viruses. In the prolactin I, we are ready to go to human trial on the cancer, and the blood substitute is the same. We are ready to go to human in this one. To summarize what we are, as a small company, we have a very experienced management in the company. We really know how to optimize the technology and the science and make it a very effective drug. We're doing in a very cost-effective, efficient development. The cost is a very important development. If you make some mistakes can cost millions of dollars if people don't know what they are doing or they're making major mistakes. Also, we have a science that proves itself. Remember, there's those 10,000 articles demonstrate a very powerful science that I'm proud to start that whole journey. And I'm leading the effort how to optimize the best drug into the clinic. And the result we had as a demonstration of the proof of concept on corona is just one case that we can repeat it again and again. So we have a very strong value inflection point in doing licensing to companies. Our business is basically, in the end of the day, is to license our technology. And the next one is to summarize the company. We are an OTC company, QB, fully reported. We expect sometimes to go to NASDAQ, to uplist to NASDAQ. And most of the shareholders in the company are retail shareholders. We have about 2,000. Outstanding shares, we have 175 million, and 53% are held by insider. Thank you.
Thank you, David. Thank you, Doctor. We have a quick question for you. Talk about if you're planning on modifying your clinical trials to incorporate some of these new viruses that appear to also be neutralized by your prolactin.
Yes. We think since the coronavirus patients population for the type of test we do, the pharmacokinetics, or how many tablets maximum we need, is kind of difficult right now because the number of patients available in the hospital are very low. We're thinking about we can go slow with the clinical trial the way we are now, or we can switch to other viruses like bird flu virus or rhinovirus, or the other virus that we show on the slide I show in the slide presentation that show very strong efficacy. So we need to modify the clinical trial by the FDA. We need to go back to the FDA, modify the trial, and expand on the patient population so we can attract a much more available patient in the hospital so we can control the trial. And that's what we're trying to do now.
Thank you for that. Now, it seems like the size of your platform technology is massive. So I'm curious, are you looking to collaborate with other companies?
Yes, that's the main direction we are looking for. We're collaborating with other companies, but officially we need to find a partner for each drug that we're developing. Basically, we have couple of drugs for each platform, but in each platform we need to find for each drug a company. We hired an expert to help us and manage those presentations. And we're developing now the presentation, the relation, and eventually will end up with a license. That's the company is all about a very narrow and very important, I would say, goal of taking a very highly sophisticated science and translate that into effective drug, what you call bed, or in the patient bed, or for patients. We demonstrate the proof of concept in a very powerful way with the corona. We have result the same, we have very strong result in cancer, and the same thing with the blood substitute. So the idea is to find the right partner, and the key is the right partner.
And Tomas Johnson asks if you could explain the timeline on the India trial, start date and time to complete.
The clinical trial right now, we need to find more patients. We are recruiting very slowly the patient on the pharmacokinetics. So that's the stage we are now. We don't know the timeline. Based on that, it may take by the end of the year.
And Lance Griffin asks, how broad is the range of treatments for your technology?
The drug that we have now is it's a broad range, like antibiotics. It's a broad range antiviral. We find out the drug right now works on at least 7 major viruses. So we need to put it in human, as we did in vitro. Since our success on predicting what will be the outcome with COVID based on the galectin the inhibition of galectin, I predict that we'll have very good data in the clinical trial with the other viruses. So it's a broad range antiviral drug. It will be a major antiviral drug. We just need to find the more viruses we find in human, the value of the drug will go up, because it will show that you can treat with the same chemistry specific, you can treat different viruses.
And last question, which is kind of a follow-up of what you just said. How effective will your product be against the bird flu, which you mentioned? And are there any emergency type grants that your company can apply for if you've not already?
We can work on the bird flu. The point is that we need to raise more money for that, number one. And the grant system is very slow. It can take up to a year or more when you submit a grant. So I don't know how fast they will react. We'll try to submit a grant, of course. Actually, we have one grant in action, but the timeline is very slow for review and approval. And the amount of money is not substantial. It's very small.
All right. Well, Doctor, thank you so much for joining us. Thank you for this update and all the hard work you're doing on your end. We hope you come back in the future with some more updates.
Thank you.
All right, everyone, stay with us. We'll be right back.